Okay, I need to. You got it. Okay. Thanks, Dave. So, yeah, this is the title of the talk. I'm going to introduce you this software platform called Annotation Query or AnnoQ. I will. Let me see how I'm going to move forward. How do I move the slide? Somehow none of the.
Yeah.
How do I do this? Do I do it on this screen or?
Right here.
Oh, I did here. Okay. I tried to. All right.
Wait, it's on your screen. So this is okay. So you have dual slides. Hold on 1 second. There you are. Now you're on the screen, but you want to do this. Hold on.
Right. I can just do this.
Yeah.
Yeah.
So this
Yeah.
is your.
Yeah. And then. Oh, now it's
So
working.
this
Okay.
think of this as your
All right,
second
all right,
screen.
right.
And this, yeah.
Yeah. Okay. Sorry about that. So this is the outline, and I'll first talk a little bit about the motivation to build this tool, and then give you an overview, and a live demo. So the project started like about 6 or 7 years ago when we started this PO1 funded NIH PO1 funded image project, which is to develop a statistical method to analyze colorectal cancer data. And we tried to figure out how to annotate, how to find different tools to annotate variants. So this is what we found at the time. There are a lot of different tools out there to annotate variants, but almost all of them require some sort of a computational and bioinformatics skills. It's very difficult to set up. Each tool will have different kind of system requirements, and different resources will have different file format. And also, you know, they support different kind of different genome build, and more subtly, and the namespace or ID space are different because they support different type of gene ID. All these kind of nitty-gritty things make it very difficult for labs to use them without the help of a core, for example, electricity. And it's not very cost effective if someone wants to annotate like a handful of variants, and you have to go through all these hassle to set up those tools. But you only need a few of those. And the other thing is, most of those, almost all of those tools, they lack functional annotation. What I mean functional is like pathway annotation or functions, biological process, things like that. Even if they have, it's very sparse or oftentimes outdated. And there are annotations to the enhancer, but also there was not enough data to link those enhancers to the gene. Thus, we don't know what these enhancers do and what kind of function they are involved. So that's why at the time we decided, you know, let's try to see if we can build a comprehensive and easy to use variant annotation system, and with the most up-to-date functional annotation, and it is accessible to researchers with different research background. And 6 years later, we have a annotation query platform. And this is the homepage. And last year, we submitted work to Nucleic Acid Research Web Service issue, and was accepted and published the paper here. And you can get more detailed information in this publication. So the overall architecture is illustrated in this diagram. Basically, there are 3 elements in this whole system. The core of the system is on the left side. Let me see if I can. Oops. Yeah, is this part, which is the database. So the way we do is slightly different from all the other annotation tools. Instead of asking you to, you know, install and run those annotation tools like SCORE, but what we do is we pre-annotate all the data. and with most of the available tools. And then we store those annotation data in the database. So now instead of scoring or running an annotation tool, what you do is simply submit a query to retrieve the annotation. And so the key of this, to build this database is really the key, is the core of this whole system. And then we have ways to access and different ways to retrieve the results. So I'm going to first tell you a little bit about how we build this database. And it's basically two elements in this database that's different from others. One is, it's an integration. We have to integrate a lot of tools together to annotate, pre-annotate. The second is to incorporate functional annotations into this whole system. So how to annotate the variant to the function, right? We know, like for decades, people study functions through the genes, and we have millions of papers describe these functions in terms of genes and gene products. And for the last 25 years, probably, a lot of expert scientists, they started to mine through this knowledge in free text and then transform them into some kind of structured knowledge. That particular process is called biocuration, right? So once this knowledge is formed, and then they are represented and stored and represented in a computer readable format, and we call knowledge base or database. So we have a lot of those information or resources available out there. But unfortunately, most of the current annotation tools, they don't connect those. So what we are trying to do is to see whether we can connect variants to this knowledge. And so the key is, it's not that difficult, because as long as we can connect the variants to gene, then we can do it. So variants can map to the DNA chromosome region, and then we can map to the gene region and intergenic region. If it's in the gene region, you connect to the gene, connect to the function. But if it's an intergenic region, we can still map to all these regulatory regions, epigenomic methylation sites or enhancers, and they can be mapped to genes and connect to functions. And there are a lot of tools actually out there. What I mean is, these variant annotation tools, they do the job for us in this area. And they also, provide allele frequency and disease information at the same time. But, we don't have a lot of information, we don't have a lot of tools that connect this directly to here. And this is usually missing. And you have to rely on different tools in order to do that. You have to do the annotation first, and then somehow process and run other program in order to connect those. So what we are trying to do is we make all these into an integrated system. And fortunately, there are resources available for us to do that. And one very useful resource is called WGSA. It's a wrapping script developed by there's a reference, Lewis Lab from University of Southern Florida. And it is basically a wrapping script to incorporate all these tools and annotate them. You still have to set up all these individual tools, but at the end, this script will understand how to run these tools automatically and do it. So we're fortunate that we can use this tool for our purpose to at least get the first part done. And we also have a lot of knowledge about how to communicate with other resources like functional annotation resources like gene ontology, Panther pathway, and reactome pathways. And this can be done through a Panther service, and we can retrieve. So once we've done the WGSA annotation, we know the gene information, and then we can use the gene information to retrieve these functional annotation data. And if we have the enhancer information, what we can do is to retrieve enhancer to gene link and annotate those enhancers to their target genes and link the functions to these enhancers. And this is through this database called Peregrine. It's part of the this P01. It's also developed under this P01 grant, this whole image program. So I want to make this as interactive as possible. If you have any question, feel free to ask. If I don't explain anything, and just ask.
Actually,
Yeah, go ahead.
this is kind of related. I think this is a really cool pipeline. Could you use it for larger features than just like single variants? If you had say differentially methylated regions or lengths of the genome?
Right now, it's only, whatever the tools they provide right now, pretty much the GSA only annotates, the version we use only annotates single variants.
Okay.
Yeah, but I think they have later versions, they can annotate like regions and methylation sites, but we haven't incorporated them yet.
Yeah.
But can you do a range?
We can annotate a range. I'll show you. You can annotate a range, but not it's still based on individual. You can give a range. It will return all the variants in that particular region and give you the annotation.
So one of the thought that, so you haven't really talked about the variance and disease link here. Is it included in
Yeah.
WGSA?
Yeah.
They pull from GWAS catalog then?
Yeah.
Got it. Okay.
GWAS, CleanVar, and Cosmic, they pull
They
all
pull
these. Yeah, all of those. And we looked, they did a really good job because if we want to do individually, it's kind of difficult because their format and everything, nomenclature or vocabulary, they use very different things. And WGSA basically is a wrapping, does it for you. So that's a nice part. So just a little bit more about the workflow, how we build it. So the variant data set we use is from this HRC data set, which contains 39 million right now as a starting point, 39 million SNP. We can expand that to, even the DB SNP or whatever, but as long as the hardware can support it, because the space it will need a lot more disk space, right? But still possible to do. And then we annotate those, like I said, first with WGSA. It has tools to provide gene model annotations, epigenomics, gene-centric, allele frequency, and disease and non-coding, a lot of others. And once that is annotated, anything that has a gene associated to those variants, we also annotate them to functional annotations such as Panther pathway, Random pathway, gene ontology. And if anything connect to an enhancer or annotate to an enhancer, then we use a paragraph to annotate to the gene also. And you can imagine, like at the end of the day, this is a large matrix, right, with a lot of rows as variants and columns as annotations. And we then build this whole data set into the Elasticsearch framework, so that the goal is basically to make it more efficient for people to retrieve the data. And these are some of the commonly used tools that are included in, for example, WGSA and Panther Service and Paragraph. For example, we call gene model-based annotation. It includes very well-known, commonly used ANOVAR, SNP-app, and VEP, and it has encode data in there. It has 1,000 genome and a lot of others, right? Okay. And here is the number of annotation types for each of those tools. For example, ANOVAR, if you use ANOVAR, it will give you 56 different type of annotations. It will give you gene annotation, effect, or cDNA transcripts, a lot of different annotations, even gene ontology, which is added by Panther Service. And you can see there are many different, so total is over 600 different annotation types. So you can go in and choose whatever you want. And this is actually the first time you can actually put all these annotations in one place, so that, and in your fingertip, basically, you can put a SNP and you get like 600 annotations. Of course, it's hard to navigate through these 600, but you can take a look. And one thing we curious at the time when we built this data set and we took a first look was, are they the same? For example, ANOVAR versus SNP-app, they are all gene model, they predict to the gene. Do they give the same results, or they're going to be different, right? Because that can affect your analysis. A lot of labs they will use ANNOVAR, and they probably think they are all the same, just use one tool. But at the end, if you use ANNOVAR, you get the results, and then you use the SNP, you may get different results because their annotation might be different. Oh, they could be the same. So we are very curious. We try to take a look, and it will be. This is actually a great place for us to do this kind of a comparison, and also if we can get information, we can pass it along to our users so that they can make educated decision how to choose these different annotation tools. So we did some kind of a pilot study and just a very rough comparison. For example, we compare the annotated genes among these three tools like ANNOVAR, SNP, and VEP. So this is what we found actually. The x-axis is the chromosome number. And for each chromosome, and the y-axis is how similar they are for each chromosome, you can see they are about 50 to 60% of the annotations, they are the same from all these three tools. But they are 30 to 40%. Only two of them, they agree with each other. And 10% of them, probably only one of them, they agree with each other. So you could get quite different annotations by using these tools. So this is something that we were surprised a little bit because we didn't think it's going to be that big. But then you can, once you look at all these on the same on the same website, and you can see how you want to use these annotation tools, and whether you want to use a union of all of them or you want to use the intersection of them. Basically, it's some kind of information I think is useful for our users, and we are doing more analysis. Hopefully, more data will be available for users to see. Okay. So now I'll come back. I described a little bit about an overview, basically, about this database part. The next part is we build a API with complex query, and so that we can, the user can access the data through this API. And there are three different ways to access this API. One is the interactive website. This is the one I'm going to give you a demo. And the second is so this one, because the way it's implemented, we encourage people to just submit a small list of the variants. It won't support large number. Imagine a lot of people are using it. If everybody submit like millions of things, I think the system won't survive. So usually if you have 10,000 or less variants, you go through this way, and you can retrieve the results. You can view it on the website. I'll show you. And if you have more than 10,000, you want to do batch annotation, we suggest that you use this API script. And I hope if you are doing like a 10,000 or a million, you have some kind of skill to run the script like that. And there's a tutorial in there. This is basically a command line script, command line thing you can enter and retrieve. But we also built some programming package so that you can embed this into your script. For example, we have a R package right now. We hope that we can have also Python package so that you can embed into your script, and you can write an R script and let it access the data. And you can, for example, running R script analysis, you can retrieve the analysis on the real time. Okay. That's pretty much an overview. I'm gonna now show you how to use this. So the URL is AnnoQ.org. And let me see. So this is the homepage, and it's no logins needed, free to use. And there's a very nice tutorial recorded by Brian. And if it's a way better tutorial than what I'm talking here. It's really detailed, and you can go ahead and take a look if you want, and before you use it, or if you have any question how to use it, you can use this as a reference. And I said we have right now we have 39 million variants from HRC and over 600 annotation types. So this is the homepage. The remaining are some of the information about this system. The interactive website is here. It's the Query UI. So, and the UI tutorial will basically tell you, what this page is about, what you can do, and everything. And feel free to use this page to get familiar with the UI. Let's see, where is it? Okay, now I'm gonna launch this UI. This is the query page, interactive query page, and the left panel is the query panel, and the right panel is the results. The query panel, there are two of them, two sections. Top is is for you to input the query, and there are different queries you can input. For example, here it's just the chromosome location, a region, so you can give like a chromosome and start and end, or you can give a VCF file. You can enter a gene product, meaning a gene identifier, protein identifier. It will retrieve all the SNPs, or you can give RSID. And we built a keyword search in there in the beginning, but there are some issues with the keyword search. I think we try to fix that problem, because when you give a very generic keyword, you can imagine the number of variants that can be returned, and we don't have any control. Sometimes it will crash the system. For example, if you enter protein, we are screwed. The system will die, right? You have to give a very specific one. So we are thinking about how we can narrow down for people to do, a more specific search. And we actually have a way to do it, but we are in the process of implementing it. So right now, please don't try keyword search. We probably should disable that, but others are all working right now. So that's the first part. You input your query, and then in the bottom part, these are the annotations that you want to retrieve. And I think if you are not, I think at least I have no idea. 600 of them. I probably only know like 10 of them or something. And a lot of them I have no idea. But there is a information button that you can click. It will tell you what it is, and sometimes there's a link and even PMID. So you can go there and take a look at what these are. What is the tool? And for example, what I'm going to do is here, I'll choose a few of those that I think I know. It's Ensemble Gene ID, and this is by AnnoQ, and I'll do a SNPF. I'll show you the same thing. It's also a effect, and in Gene ID, and I'll also select a VEP. And VEP is not called effect, they call it consequence. And the gene ID, and I'll give some functional annotation, for example, Panther. And Panther, basically, it will annotate functions, but depending on how close the variant to the gene, it could be only allow 0 flanking region, or sometimes we can allow 10,000. So let me see. I'll put a pathway in here, and maybe a molecular function. And since Dave mentioned about disease, let me see if we have the GWAS catalog, and I think this trait is the one that has some kind of name. So we submit. Now it returns. So in this region, basically the system say there are 6773 variants. And here are all the variants. And if you click one of them, because it's hard sometimes if you have a lot of columns, it's difficult to see. So you can click and you can see in a table view, and you can even click those links to, to the DB SNP or those genes. And it'll say, you know, upstream this. The reason the gene lists twice is because they index based on the transcript. Even though the transcript is not one of those you search, but AnnoVar annotate up to the transcript. So if a gene has two transcripts, it will list it as it has two transcripts in that particular column, and then it has to have, you know, corresponding genes to those transcripts. That's why it listed twice. And these genes are upstream, and this gene is downstream. And you can see AnnoVar ensemble give two genes, and SNPF, it's pretty much two genes. This is, I don't know, they put a chromosome start in here. I don't know why, but it's basically two genes also. But it uses a little bit different vocabulary, like one is upstream, downstream, and here SNPF say downstream gene variant. And VEP is the same, it's say, you know, downstream gene variant and the genes. So basically that's how the results will be displayed. And now just show you a little bit here. Like for example, here, for this particular variant, right? And SNPF will annotate to a gene. It's an intergenic region. It's probably in between these two genes. It will annotate to these genes, but the other two, they are more conservative. They don't annotate this variant to those genes. So when you use those tools, you will see the differences, right? And you probably noticed that there's a number underneath the column name. This tells you how many rows has an annotation in this column. And so, for example, here, AnnoVar ensemble gene ID, it only has 57223 genes because there are some blanks. And you can click this, it will filter out any rows that's empty in this particular column. And you can do that. And of course, you can remove this filter. So there are two kind of features you can use here. One is filter, right? And you can filter from here, or you can filter from here. And if you do a filter here, you can search the field, which field you want to filter. And there are numbers here, just tell you, you know, for this particular column, how many rows has annotation. And if you do the pathway and it'll filter, you see the pathways. Yeah, here. This happened to be in a Huntington disease gene. So all of them, they are annotated to Huntington gene. And the other feature is a stat. So you can gather statistics. For example, if you click, it will show you some sort of, it will tell you, you know, majority of them, they are in the intron region. This particular region is in a lot of them, they are in the intron region of genes, but they are also in other regions, right? You can do that. And sometimes I'm curious about, for example, pathways. I can do that. So that's how I found that, like, almost, you know, a lot of those, they don't have any, it's still listed. We should probably remove that. But 192 is involved in Huntington disease. That means there's probably one gene particular in there, all these variants map to that gene, and that gene is involved in this pathway, right? So this is basically how you can view those data, and you can filter. And the other function is you can download them after you do the filtering and all these things. You think this data set is what you want, you can click the download and download the data set. The download is not, doesn't work instantly. It's not like normally you click, it will give you the results right away, because depending on how many rows, usually it has a lot of data in there. So the way we set up this download mechanism is you click the download, then it will give you a link, and you click that link to download. If I click download, it will take like a few seconds, sometimes a minute. And then it will give you this link. It's temporarily stored in our system, so that now you click this and you can download the file. But I think, I don't remember the time. It's probably at least an hour or two. In that time frame, you can come in and download it. So you can download the results, and it's captive made, I think, file. All right, and now let me show you some other queries. So the second one is the VCF file. You can upload the VCF file up to 10,000, of course, in here. And you can retrieve annotation results. Here is an example VCF file. We can just load it from here. So one thing I want to caution you is, this VCF file format. So we noticed that in some VCF files, like in the alt residue, there could be more than one. You can have a C and a G. The format is not consistent. In VCF file, you can put a C/G or C,G or whatever. Unfortunately, for AnnoQ, it has to only have one residue in here. So if you have multiple residues, you can split them into different rows, or because it doesn't know whether CGGC is the same. So that's why it's a kind of a weird system. But so just remember, you have to use only one residue in this alt column. And I can submit. Now it will return the results, and it will annotate a total of 2005 variants from this VCF file, and give you all the annotations that you pre-selected here. Oh, yeah. Now there's another thing I want to mention here is the configuration file, what we call it. So when you select the annotations you want to retrieve, right? You will click these boxes, and sometimes it takes time for people to decide what to annotate. They'll go through different multiple iterate in order to see what kind of an annotation they want. At the end, they decide a set of annotations they want to retrieve. But imagine you do the analysis, you retrieve the data. A week later, you come back, you could completely forget, at least I won't remember what I selected a week ago. So I have to go through this process again. What this nice feature is, actually you can export whatever the annotation type you selected from here. And you can save it as a file, as a configuration file. Once you save it, next time when you come back, you can just upload this from this file, and then it will give you exactly the same selection of annotation. Let me see if this works. So let me remove all the annotation types I selected now. Okay, oops. Now this is a configuration file I saved before I open. Now I have all the annotations selected for me. Just imagine, this is going to be very useful, because imagine like you are collaborating with someone and you did an annotation. You don't have to tell him, what I selected. You just send that configuration file, and they can run that similar analysis on this with different data set, but exactly the same annotation type selected, right? Or you want to make a publication. We run the analysis over and over, and you can make sure that you always use the same set of annotation data. So now this is the VCF file, and the next is the gene product. This is actually a suggestion from Josh. You suggested it, and actually it's a very useful query. That is, you can enter a gene product ID, and after you enter that, you can search for variants that are mapped to this particular gene by any tools, and retrieve whatever the annotation you want from this annotation tree. I do this, and I found that this particular gene is in chromosome 10. So here on the top, it will tell you, this is mapped the gene to the Panther database. This is the Panther database ID. Don't worry about it. And it will tell you there are 585 variants that are in this gene region. and this gene is in chromosome 10, and this is the region. And it will return all the variants within that region. And plus all the annotations that you select. Okay. And the next is the RSID. RSID, you can do single ones or a list of those. This is, again, we found it very useful. The reason is because, for VCF file, there is a genome build information embedded in there. So you have to know, and if you happen to not having the same genome build, now you have to convert the chromosome location and all sorts of things. But if you have the RSID, that's basically genome build independent. You can just use the RSID and enter it here. You don't have to worry about all those coordinates, right? So here's an example. Let me populate one that I got from Jim. This is a better one. I hope. Is it okay to show this in? Is it okay? It's recorded, and I don't know. It's okay, right? Okay, I populated this, and I submit it, and these are the results. And there are 203 variants in this list, and it will tell you, what each one is. Just for fun, for example, I did some statistics. Let me see. For example, the pathway. I just want to see what kind of pathway these variants are annotated to. And a lot of them, they don't have any annotation, right? But there are 5, like in TGF beta pathway and, integrating and also Alzheimer's disease, all sorts of different pathways. So that's kind of interesting. That's something you can, when you do the annotation, you can get results like that. And that hopefully, these are useful data for you to, to guide the analysis for the analysis. Let me see what else I want to cover. Yeah. So now come back to I mentioned about the configuration file earlier. So another thing I want to mention is, when you create a configuration file, there are multiple places you can do that. You can do it here, and you can go through this list. And there's also this place, supported annotation. And it's a more user-friendly interface. Not only that, you can search for terms because there are 600 of them. If you just go through the list, it's difficult sometimes. So you can do actually search here. For example, ensemble. Oops. Yeah. So if I click ensemble, if I type ensemble, and now it will show all the ensemble related annotations, and you can narrow down and you can make the selection. And if you do like effect, hey, what happened? I don't know. Probably. I don't know, something. Probably. Anyway, you basically can do these kind of a search from here and look at the list and decide what you want to select. Once you narrow down the type that you want to select, and you can, export that configuration file. Go back to your website and upload it into that to run analysis. So that's pretty much what I'm going to say about this interactive web query interface. And there's a detailed tutorial here also. You can use that to get to other ways to access the data. For example, use AnnoQ API. And if you click here, it'll show you, give you some tutorial about AnnoQ API, how to use that what kind of a, how to construct a short query to retrieve the data. And there is a link that will show you some example bash scripts. So you can take a look, use that as a template to write some kind of short script, or you pretty much sometimes you can use that just straight to change the number or whatever. You can get the results. And there's also a AnnoQ package, how to use that, and there's a GitHub repo with some scripts, sample scripts, so that you can use that as a template to do that. Let me see. How do I get back to my slide? Okay. Up here. Okay, I also want to advertise, not advertise, just tell you that previously we did a Happy Scientist seminar also, it was 3 years ago. In that particular one, if you go to YouTube, go to Biostat seminar, you will find that AnnoQ presentation from 3 years ago. In that presentation from minute 24, there are real demo about how to use those scripts to do, API access and the R package. So just, I have very little knowledge about those R scripts and how to use them, but there are some real nice demo in here. You can take a look if you want to use that. And the website is pretty useful also, and there are scripts you can use them as template. So just to recap some of the nice features we have, one is the most updated functional annotation. We have integrated that into the regular annotation, variant annotation pipeline. And actually those functional annotations are most updated. We try to keep them updated as much as possible. They are updated in Panther service on monthly basis. But we cannot do that. We cannot afford to do that. So we are trying to do it every 6 months to update the AnnoQ with a more current functional annotation. The other feature is configuration file. I mentioned, they can provide you with consistent annotation data sets, and it facilitates the collaboration and publication and result download, and you can use the download results from even the website and all the other ways to retrieve the data. And coming back, this is basically, again, a summary of our system, right? We build a annotation database and a API with complex query to access the database. And then there are 3 different ways to access this API to retrieve the data. And for the interactive one, we can save the results from the website, or we can also download a configuration file. These files can be used in the other ways when you use other ways to retrieve the data or annotate the data. And this is a summary. I believe, I don't know if I convinced you, it's a pretty easy to use. It's a website. It's just a query, right? If you can do Google search, this should be the same. And you don't need to set up anything, and you don't need any computer. As long as you can do Google search, you can do that. And we have multiple ways to annotate, and you can use the web interface, or if you are more skilled, you can use a programming package such as R or API scripts. We provide comprehensive functional annotations, and we actually, for the last couple months, I've been working with Jim on his data sets, trying to see, how the functional annotation will help his analysis in this Fiji data. And hopefully we'll have some results in the near future. And also I didn't say too much about it, but we also incorporate the enhancer function annotation in there, which is actually a very useful tool, so that you can actually add quite some more functional annotations to variants that are in the non-coding regions. And we are very happy to notice this, see that. So here are the people I want to thank. So this is the AnnoQ team. Tremaine is the one who right now is the main person who is maintaining this system, this website, and with a lot of help from Brian, who is a student, Biostat student. And we got a lot of engineer help, software help from Anisha and Dustin, who's here. And the project initially was, it was started by Zhu, who was a student here also. A lot of ideas in this system was originally from him. And he left, he graduated, and he went out to some other positions. And Caitlin is a student in Biostats. She's the one who developed the Peregrine database. And I would like to thank everyone in the image team who provide a lot of wonderful feedback along the way through the last 6-7 years. And I hope that this tool will be useful for you and with to all the others, and we'll continue to work together on this. And of course, I would like to thank a lot of other people who provide all these useful resources for us to develop this tool, and it's funded by the PO1 grant from NIH. If you have any question, you can ask right now, but if you have any question afterwards, this is the email, annoqfeedback@gmail.com. Just send email and I'll try to, or someone will try to answer your question. But feel free to stop by my office or email me, just, if you have any suggestion or question. Thanks. Any question?
Okay, I have a question. Can I? Thank you. I think very useful this software, but I'm totally new to this field. So I'm wondering. So of course, to use your tool, you need to have the variant file first. So any software you can let us know which which softwares we can use to firstly retrieve these variants. Also, for now we have single cell sequencing data, bioRNA-seq data, and or we can do the DNA sequencing. So any difference for those data when we analyze them for the variant?
So what you are asking is, if you have the sequencing data,
Yeah.
how do you know it's a variant, right?
Yeah, which softwares you can suggest to use to, firstly, we need to retrieve, analyze those, to find, to discover those variants.
So I'm not sure. There are tons of those, right? And Dave, you probably know better about what kind of a.
Well, yeah, I mean, I think variant discovery is one thing through sequencing, like germline. I mean,
Okay.
the key here, though, is to use in this database. It's from the haplotype reference consortium, so it's limited to the 39 million SNPs that have already been discovered and called from that data set, right? And
that's true.
And
so
yeah.
So so you're saying that called variants, and there are tools to do that. There are scripts. I'm not 100. I don't know if I have a good answer to that, but I know there are.
GATK.
Yeah, GATK, and I heard about those tools, but I never use it. But your question about limitation, yes. So the limitation of this tool is it limits to the number of variants we have in the database, and it limits to the columns, basically the annotations. If you want anything outside of that, you cannot do it. So the annotation type, I have to say, it's pretty comprehensive, because the 600 column probably covers almost every tool out there. They are doing whatever they are doing. But I agree that 39 million variants is probably not enough. And we started this was because we were doing the Fiji, we were working on this PO1, and a lot of data is from the Fiji project, which is from the HRC, right? And we actually annotated the top map and the DB subset of the top map, and we could, you know, incorporate, but like I said, there's a hardware requirement because the disk space has to be 20 times, no, not over 20 times. It has to be like maybe 80 times bigger. So we are working on that. So we'll probably, but of course, and you can still argue that there may be other variants outside of that. So that's true, but the limitation is whatever included in there, that's what you're going to get. We believe that if we can incorporate TopMed, we should cover almost nearly everything. Yeah, Josh.
Can you explain the keyword search again? So you enter, like, what kind of keywords can you enter?
So the keyword search originally, we thought, you know, if you enter, for example, TGF beta pathway, and all the genes that have been annotated to TGF beta pathway and their variants will be returned. And but if we allow this kind of free text search, the system is not going to work. We started and prototyping, it worked with a small set. But once we have the entire 39 million data loaded in there, we noticed that it was slow. So like I said, it's basically partial match. You can match one term and then retrieve like tons of genes and then the variants. It's just not going to work. So what we try to do is we try to, just like what I show you, like when you search the annotation type, if you type a term, it will give a choice, a list of choices, different terms that's in there. And you are forced to select the term in our system so that it's not a free text. It's just a specific term. When you select that, we'll only retrieve those. That probably will work a little bit better, but it won't give you that kind of flexibility to search everything, but at least, you know, it won't crash the system. So
what type of terms?
the term will be like a G protein coupled receptor signaling pathway, for example.
What
Or
about a disease?
you could search a disease, but once you type a colorectal cancer, it would only, you know, you type colorectal, it'll only say colorectal cancer. And you select that term, or is there any other colorectal, whatever, hemorrhage, or, and then you can only search the term that's listed in there. We basically force you to search the term that's in our database. So we don't have to do a partial matching.
Sounds great.
Yeah, we hope that we can implement that. And that will allow you to do. And I think the reason we incorporate this keyword search in the first place was there were requests, I don't know from you or from someone, say if they want to enter a keyword like a disease, right? Or P53 pathway, they want to retrieve every variant that's possibly involved in P53 pathway. So that's why we thought about it. We think it's a good idea. We implement that, but we still have to test it. Once we update our keyword search capability, we still want to test to see if the system will survive with that kind of implementation. Thanks.
How big the engine that you're using now?
How big?
Yeah, how much is space, how much time?
So
Is it part of the HPC?
no, it's hosted here in Bioghost, on Bioghost, and Bioghost 2 specifically. So right now the machine, the data is a few, I don't remember exactly, but a few hundred gigabytes.
Mm-hmm.
So not quite a terabyte, the data in there, and probably it's 500 gigabytes. So if we want to host like a TopMed, the data will be huge. But we actually identify some areas that we can optimize the disk space, and that's another thing we want to do. It has nothing to do with your search or whatever, but it's just,
Yeah.
we have some waste space in there.
So are you doing a lot of indexing?
Yeah. So the data, we started with the data like, like a large file format, and then we converted it into JSON format, and then index with this Elasticsearch thing.
There's something in the chat, maybe.
Oh, let me see. Oh, yeah, there's a. You have to update these. Yeah, so basically, we do.
Read the question.
Yeah, so the question is, do you plan to update these background databases with the time which you are using, like AnnoQ, VEP, and SNPF? So I guess is how often we are going to update those, right? So, so that we basically rely that on WGSA. WGSA, they basically update between 6 months to a year. Sometimes they had a longer gap, I guess, probably funding issue or something. And each release, they will tell you what version of AnnoQ, SNPF, and VEP they want to use, and they will support those new versions. And AnnoQ and VEP, they actually change format sometimes. So if you download the old version and run the WGSA, sometimes it doesn't work either. So they have specific version of the tools they support. So it's kind of a complicated process. So every time we update this WGSA, rerun it, we have to download all the tools again, install them, and then run this WGSA. And we try to do it based on their update schedule. And if it's 6 months or 1 year, so we'll just do that. And we probably won't do it shorter than 6 months, because that's a lot of work.
Do you, in your configuration file, do you have that information?
So that's another thing. The configuration file is version specific. If we update a version, then you have to have a new. I don't know. That's a good question, actually. How we can update, archiving the old version and migrate this to a new version. That's a really good question. We haven't thought about it. We haven't really done anything. And archiving is really a question of again, a disk space issue. It's not very difficult to have an archived version, but we just don't have enough space to archive.
All right, well, thank you.
Thanks.
Yeah.
So,
Yeah, if you have any other questions, just send feedback or